Hi everyone, welcome to Bioorganic Chemistry. In the previous lectures, we have studied what are monosaccharide, their structure, structural transformation, application, and some of the properties, including chemical reactions. Today, we are going to look at disaccharides and polysaccharides, their structure, property, and some of the applications with representative examples. Disaccharides are carbohydrates which have two monomer sugar units which are linked via covalently through a glycosidic linkage. We will look at some of the examples such as cellobiose, lactose, maltose, and sucrose. Cellobiose is a disaccharide that's obtained by the hydrolysis of cellulose. It contains two glucose units which are covalently linked via beta 1,4 glycosidic linkage. This beta linkage, because of this glucose, particular glucose has hydroxyl position at the anomer carbon in the equatorial position, and the carbon number 4 of the other glucose unit is linked. So this is the glycosidic linkage that we see here. This can be broken down into two glucose molecules by acid or enzymes such as beta glucosidase. Lactose is another disaccharide that contains a galactose, a D-galactose, covalently linked to a D-glucose unit via once again beta 1,4 glycosidic linkage. Lactose is commonly known as milk sugar. It's a carbohydrate from the Latin word lac, meaning milk. And it's important to know that many people also suffer from lactose intolerance. In the small intestine, the enzyme lactase breaks this lactose into glucose and galactose, and these monomer or monosaccharides are then absorbed in the bloodstream. So people who are suffering from lactose intolerance, which is a common digestive disorder where the body is unable to fully digest lactose due to lack of lactase enzyme. So there are more than one type of lactose intolerance. The first one is primary lactose intolerance, which is the most often found lactose intolerance case where the lactase production naturally declines as a person ages after infancy. The secondary lactose intolerance can occur due to an illness such as celiac disease or Crohn's disease, or surgery that damages the small intestine. There is one rare case which is congenital or developmental. These cases are when infants are born without lactase or premature babies who have temporary lactase deficiencies. We also have to remember that lactose intolerance is not the same as a milk allergy. A milk allergy involves the immune system and can be life-threatening, whereas lactose intolerance is a digestive sensitivity. Maltose is a disaccharide. If you look at it, also has two glucose units linked together. However, the linkage, the glycosidic linkage, is now alpha because the hydroxyl group at the anomeric position is in the axial ring. So this is an alpha 1,4 glycosidic linkage, and maltose is commonly found in plants and many other plant products, barley, corn, wheat, and also in malted products such as beer and some fruits. This can be obtained by hydrolyzing amylase, one of the components of starch. Enzyme amylase can break maltose, amylose into maltose. And further, an enzyme called maltase can break this into two glucose molecules that will be used for absorption. Sucrose, commonly known as table sugar or saccharose, contains a glucose and fructose linked together via glycosidic linkage, which is alpha 1 and beta 2 glycosidic linkage. Now, if we go back here, all the previously discussed disaccharides, there is, in this case, it is an acetal. However, this end is a hemiacetal. So, therefore, this can still be used as a reducing sugar, whereas The end of this particular side, in the left side, this is non-reducing end, whereas this side is a reducing end. So most of the disaccharides that we have seen until now are reducing sugars. However, in fructose, both of the anomeric carbons are used for glycosidic linkage formation. Therefore, this is an acetal. There are no hemiacetal. Therefore, this is a non-reducing sugar. Polysaccharides are carbohydrates which contains a large number of repeating monomeric unit or monosaccharides linked together glycosidic linkage once again. And some of the common examples are cellulose, starch, glycogen, other polysaccharides such as alginates, agarose, and chitin. So some of the properties of polysaccharides are they have very high molecular weight because of the large number of repeating units. They always have a glycosidic linkage. They are interconnected or covalently connected by a glycosidic linkage. They have repeat units. These repeat units can be of the same type, then they are homopolysaccharides, or they can have a different repeat units, then it can be heteropolysaccharides. They can be linear or they can be branched. The branches can be long and few or short multiple branches. So other characteristic, as I said, they are insoluble in water, they are tasteless, they store a high amount of energy, they are often as a structural supports such as cellulose in plant cells. Examples, as we already indicated, starch, cellulose, glycogen, agarose, alginate, and chitin. Let's begin with cellulose. It's one of the most abundant biopolymer in the earth, which provides a strong structural support in plants from grasses to trees. It is essential in the human for digestive health and for food movement and elimination of waste. Carbohydrates such as cellulose can be broken down using hydrolysis into glucose unit. They can be further used to create biofuels like bioethanol. Now, majority of you might have seen the structural representation of cellulose as a polymer with the repeating unit of cellobiose. However, this is not true. The reason is cellulose is a long chain. It also has a non-reducing end and reducing end. Therefore, the correct way of representing is this one, where we show at the center a glucose repeating unit and two other units which indicates the non-reducing and reducing end. Whereas if you write like cellobiose, we cannot indicate reducing and non-reducing end of this structure. Therefore, the currently accepted structure is this, which shows a glucose unit at the center and the two other. Cellulose is insoluble in water. It's very strong, and the reason is that it has a lot of chains which form very strong intramolecular hydrogen bond as well as intermolecular between the polymer chain hydrogen bond that keep them very intact and the structure integrity. Now, if you look at here is a schematics and also some of the chemical structure and microscopy images of various celluloses. If you look at the secondary cell wall, break them down, you will get some fibers of millimeter to micrometer diameter. You can further mechanically digest them into fibrils of micro to nanometer. And in that case, you see two regions in these fibers that are crystalline and non-crystalline disorder domains. Using controlled hydrolysis, you can obtain two types of product. One is a nanocrystalline product. It's a nano because the diameter is about 18 nanometer. Now, if you look at the length, about 200 nanometer. These are crystalline, nanocrystalline cellulose. Then you can also get a long fibrillar structure, which are called cellulose nanofibers. Diameter is same around 18 nanometer, but the length can be indefinite. They also have multiple chains stacked together, and then they have certain repeat units, and then they also have reducing and non-reducing end. But the plant cell wall also and plants contain, in addition to cellulose, they also contain hemicellulose and lignin. So if you look at the scanning electron microscopy image of nanocellulose, you will see highly entangled fibril network. And because of this entangled network, they can immobilize a lot of water. This leads to a hydrogel, which we call colloidal gel because the dimension of these fibers in the colloidal size range, which can be used for various applications, biomedical, wound healing, and developing some implant-like structures. So here's a beautiful review that shows the evolution of cellulose-based material from timber, paper, packaging, membrane, bioengineering, and the future applications such as bioplastics and bio-based optoelectronics. Here is another example where we combine the left side, the genetically engineered protein, with cellulose nanocrystal to make a strong and tough material such as a dental implant structures. So that is the beauty of cellulose, where the applications are emerging all the time. You can produce one more type of cellulose, as cellulose can be obtained from many sources. This is a bacterial cellulose. If we feed bacteria of certain species or strain, this can convert glucose into cellulose, and you get a beautiful nanocellulose with a nanofibrillar cellulose in the form of membranes. I discussed about cellulose nanocrystal, as you can see in the transmission electron microscopy image of nanocrystals here. And it can disperse depending on the surface property. They also have reducing and non-reducing ends. Some of the hydrogens are replaced with the off-sulfate ester. That gives a water dispersibility. But if you increase the concentration, they start to phase separate due to a chiral nematic arrangement of these nanocrystals. It gives a beautiful color. This can be tuned by many ways by changing the concentration, the pitch height, and helical pitch. Because it forms this kind of helical arrangement, you can nicely tune the color by tuning the concentration or type of cellulose nanocrystals. And this kind of color is also found in nature. The one previously shown is that how you isolate and then make this kind of structure, but nature itself has certain fruits which have this high reflectivity due to helicoidal nature of the cellulose nanocrystals present in their structure. You can also identify why it is, because cellulose has a very high refractive index of 1.53 when it's dried. Now, if you take the structure of this, look at the electron microscope, you can calculate this pitch height and pitch length, which is about 200 nanometer in these fruits. If you now multiply refractive index with the pitch length, you get about 445 nanometer. That corresponds to the color that we actually see. And the reflectivity of this fruit is 30% of that silver mirror, which is one of the highest reported reflectivity for a biological material. And you can also take cellulose and modify and replace some of the hydrogen here or OH with ether linkages such as methyl. Then it becomes methyl cellulose. Unlike cellulose, it becomes water soluble because now you eliminate the strong hydrogen bonding formation between the chains. And this forms a very interesting type of gel. Normally, when you take a solution in heat, it becomes very thin, but viscosity should reduce. But in this case, if you take the dispersion of methyl cellulose, which is one of the widely used viscosity modifier in the food, we eat it every day in one or other form. If you heat it, it forms a gel. It becomes stronger, viscous, and stiff. And this particular property is called thermosensitive gelation. And that's because when you heat, they expel water, they start to coil, they form fiber-like structure. This can be visualized by freeze drying this kind of gels and just drying this kind of gels which are not having fibrous structures. When you heat, you get this fibrous structure, and you can also use cryo transmission electron microscopy to visualize individual fibrils. Because these gels are now shear thinning, that means when you apply force, they start to flow freely. You can use them to extrude something like fiber or make mold using some coagulant bath, where coagulation is a process where it will solidify these kind of gels. Remember, this extrusion involves 96% of water in the gel, only 4% solid or even less. And then if you use isopropanol or ethanol, it coagulates or solidifies the fiber. Therefore, you can do a continuous fiber extrusion Using simple cellulose derivative, you get very strong and tough fiber. These are used as bio-based optical fibers. Other important carbohydrate that we take every day is a starch, which accounts for more than 50% of our daily intake. Starch contains two polymers. It's a mixture of two polymers, amylose and amylopectin. Amylose accounts for 10 to 30%, which is here shown a linear polysaccharide. A branched polysaccharide, amylopectin, accounts for 30 to 70 to 90%. So amylose is a branched Amylose is a linear polysaccharide where we have glycosidic linkage, which is alpha 1,4 glycosidic linkage. This is water soluble and it can be easily hydrolyzed into glucose units by enzyme alpha amylase. Amylopectin, on the other hand, has two types of glycosidic linkages, alpha 1,4 glycosidic linkage, and then again this branch structure alpha 1,6 glycosidic linkage. This is not water soluble. Now, there is a beautiful test to detect starch in any food item or fruits or any other substance. This is using starch iodide test. And this is a very old test where you use iodine solution or I3 minus, like a mixture of iodine and potassium iodide. That will give a very dark blue color. This test was developed more than 200 years ago. And this is because when you have a starch and a lot of amylose content, amylose forms very nice helical structure and it forms a complex with I3 minus that gives a dark blue color. Glycogen is another branched polysaccharide and it is the energy storage carbohydrate of animals. And unlike amylopectin also had a similar repeating structure or branch structure. This has a large number of branches. However, the length of these branches can be short from 8 to 12 glucose units. So that is the main difference. Super branched glycogen and branched but longer chains, less branched but longer branches in amylopectin. The another important polysaccharide which we use daily is alginate. Alginates are extracted from brown seaweeds or some bacteria. These are linear polysaccharides, but they have two types of repeating units. The property of these changes based on their repeating units, which are like G, which means glucuronic acid, M means manuronic acid. And this can be used very nicely to make metallogel because when you add metals such as calcium, it forms this egg box like structure, readily crosslinks, forms nice gel. And this has been used to demonstrate this edible water bottle using biodegradable polymer. Here is a beautiful example showing that you can make edible water bottle just by using alginate. It essentially has a very thin transparent membrane of alginate and the water is inside. So because if you have a food grade alginate which is commercially available in food stores, you can do this using some very clever way of making these balls. This can be done at home. However, you have to use food grade materials. And we can also use these alginates and related products such as with calcium or other metal ion crosslink to make gels. As I previously indicated, many of these polysaccharide gels have shear thinning property. We can modify the viscosity. By modifying this viscosity, we can control how fast or slow we can extrude this, and we can also control the resolution, like how thin structures we can make. This is a 3D printing of human ear like structure using this Agarose alginate based gels, and it is very rapid. It doesn't take much time, but you can make any type of shape because the shape is very robust. Agarose is another type of polysaccharides, which contains D-galactose and 3,6-anhydroyl galactopyranose structure. And it is commonly present in seaweeds. It is also a major component of agar. And if you take agarose and dissolve in water at extremely small quantity, you can make a transparent to translucent gel. If you look at the cryogenic transmission electron microscopy, you will see these small fibers. They are responsible for entangling, as shown in electron microscopy here below. This is scanning electron microscopy or atomic force microscopy image. They can capture and immobilize a lot of water. Now, agarose, what happens if you take a solid and add water and heat it, it forms a nice viscous liquid, and then you can pour it on any type of shape or mold. You can make mold them into different size. You make this gel electrophoresis. It can be highly transparent or translucent gels. That is because when you heat and allow it to cool down, it forms this double helical structure. They cross-link and they can trap a lot of water molecule, preventing the mobility of molecule. These agarose and related gels can also be used for tissue culture and tissue engineering applications, such as shown here for culturing patient-derived explants from breast cancer tissues. This can be used to retain their original properties of the tissues, and including then drug discovery, drug testing, and patient-specific drug identification. One of the most interesting polysaccharides present in the nature is chitin. And chitin is a long chain polymer, very similar to cellulose, but the exception is that it is made up of N-acetyl glycosamino units. It's a glucose unit which is N-acetylated. Other than that, the repeat units are very similar to cellulose. It's very tough, it's a natural, and that forms a hard exoskeleton in some arthropods, insects, and crustaceans, and the cell walls of fungi. It's used commercially. It is often extracted from shellfish, and its use is mostly for medical, wound healing, drug delivery, and other agriculture, such as plant defense purposes. One other category of carbohydrates is cyclic carbohydrates. These are known as cyclodextrins. They have a multiple number of glucose units varying from 6 to 8. When the number of glucose units is 6, it is called alpha cyclodextrin. When it's 7, it's beta. When it's 8, it's gamma. Interesting property here is that the interior of this can be slightly hydrophobic. That means if you add cyclodextrin to a water and have a drug that is less water soluble, this can encapsulate the drug molecule and make it more water dispersible. This has beautiful internal structure of pore size varying from 0.5 to 0.9 nanometer, and that can be used to encapsulate some metal ion, can be used to extract toxic metal ions or toxic dyes from water, and as a result this can also be used in membrane filtration.